Hi guys, it's Miss Enfield here, and today I'm going to show you guys how to do a cheek cell lab. So we are going to get some DNA from our cheek cells, and we are going to be able to see our DNA with the experiment that we're going to do today. So we might be able to, or you guys might be able to do this experiment at home. There's not too many supplies that are needed. They're listed on teams, but I'm also going to tell you guys what they are right here. We need some dish soap, some salt. You can just use normal kitchen salt. Isopropyl alcohol. This you might not have at home. So if you don't have this at home, you probably can't do this experiment. But if you do have this, I bet you have everything else that you would need. And then you're also going to need some food coloring, a Dixie cup or a small container to put stuff in. This right now has my isopropyl alcohol with just a few drops of food coloring, and then I use a toothpick to stir it up. And the isopropyl alcohol works best if it is chilled. So if you are going to be doing this experiment with me, go ahead and put this in the freezer for now, so it gets nice and cold. Mine was in the freezer overnight, so it's still pretty cold. But if you are doing this with me, go ahead and stick this in the freezer, and it'll give you a lot better results. And then I have another Dixie cup that I use to gargle my salt water, which we'll get to in a second. And then a pipette. If you don't have a pipette at home, you can probably still do this experiment. We're going to use a pipette in a bit to pipette our isopropyl alcohol onto here. And the way that we're doing it, it just needs to be very, very gentle, so we're not disturbing our DNA in the Dixie cup too much. So if you don't have a pipette, you can try pouring it, or maybe using like a syringe or like turkey baster type thing. You could try and use that. You can get creative with it. But if you don't have the supplies at home, that's alright. You can just watch me do this exercise. So our first step was to put a centimeter of salt into our Dixie cup. So if you don't have a Dixie cup, that's okay. You'll just put a good amount of salt in a smaller cup. So I filled it up to about there, I guess like a finger length of salt. And then I put warm water in it, and that helped dissolve the salt. And you can see how much water I have in here. It's not a ton of water. And then what you do is you gargle it for a full minute. So I put the salt water in my mouth, I gargled it, and then I spit it back in here. I already did that. You can see my timer right here. I already did it for a full minute. It's very salty. It was not super enjoyable. That's alright. I rinsed my mouth out with some coffee. So once you have a centimeter of salt water in here, you dissolve it with warm water and you gargle it for a minute. You're going to set that off to the side. And next you're going to take 1 drop of dish soap. We don't need a lot of dish soap, just 1 drop. So I'm going to drop it in here. I'll get it so you guys kind of see what I'm doing. Here I'm going to just put 1 drop of dish soap in. And boop, that's all we need. And I'm going to use a toothpick to stir it up. And while I stir it, I don't want to make any bubbles, so you don't want to stir super aggressively. We're just going to, this camera, just going to stir it gently around. We can kind of see the soap mixed in there. There we go. I'm just going to stir it around until that soap looks like it's good and mixed up. I can kind of see some blue soap pieces still spinning around. And this salt water right here is supposed to have my DNA in it because I swished it around my mouth for a full minute. So we are going to see if we can visibly see DNA. It's going to be kind of gross. You get to see Mrs. Enfield's DNA from her cheek cells. I'm going to keep stirring it for a little bit longer until I don't see much clumps of soap, once it looks like it's pretty well mixed. And again, I don't want to stir it too aggressively to have air bubbles in it. I'm going to stir it a little bit longer. You can see some clumpy stuff there on the side. Not sure, maybe that's just soap. Who knows? Alright, I think that's pretty well mixed in. So next I'm going to take my isopropyl alcohol with food coloring. So I added this food coloring in with our isopropyl alcohol. This stuff right here. There you go. And we're going to just put 2 pipettes of it into this solution here. And we're just going to gently put it on top. We don't want to like inject our DNA solution with isopropyl. We just want to set it on top gently. That's why we're using a pipette. So if you're doing this at home, you're going to want to use like a turkey baster or something like that. You could try and just like lay it on with a spoon. You can get creative with it. If it doesn't work, that's okay. You'll be able to look at my results. So I have my isopropyl alcohol solution here with food coloring in it. I'm going to take 2 pipettes. And again, this was chilled. It works a lot better when it's chilled. So if you have yours in the freezer, you can go take it out now. I'm going to squeeze one, fill up my syringe, or my pipette, and then I'm just going to gently put it on top. Alright, there's one pipette. Now I'm going to do a second pipette. There we go. And again, the same thing. There we go. And now we're going to let it sit for 2.5 minutes. So there we go. And I'll pause the video and I'll let you guys know when it's sat that long. And you guys can see I left my toothpick in here, but I'm not going to stir it. You do not want to stir it right now. That's why we put our isopropyl alcohol solution on top very gently, so we don't want to disturb what we have in our Dixie cup here. So just do not stir it. I'm going to use my toothpick in just a minute to try and show you guys the DNA clumps that form. But do not stir it. Just leave it in there. If you left your toothpick in there, that's okay. Don't touch it. Just leave it for now. Alright, so as you guys can see, our 2.5 minutes are up. So this solution of our salt water that we gargled, we added the drop of dish soap, and then we added 2 pipettes of the isopropyl alcohol with just a little bit of food coloring in it. So this solution has sat for 2.5 minutes, and now we should be able to see clumps of DNA when we monkey around with it. So I'm going to take this right here. Let's see what we can find. Alright, so it might be kind of hard for you guys to see, but there's some stringy things in here. They're kind of blue from our food coloring. They're kind of all towards the top, but the stringy pieces that we see, maybe you can see it, they're kind of sticky. But that is our DNA, and they're kind of small, of course. Trying to get a good stringy one for you to see. It's moving away. Let's see, maybe you can see some goop on here, on the stick. Maybe not. But there is, in fact, DNA in here. It's very small, kind of gross, a little stringy. You guys can also do this by crushing up some strawberries and then filtering it out. If you wanted to look up how to do that experiment, you're welcome to. I know strawberries have a lot more DNA than just when we switch it around in our cheek, so it gives a better visual. But we can see all these little tiny clumpy things, and those are our DNA. So maybe you see, there we go. I think you guys can kind of see that. The stuff right on the edge of the stick there. Looks kind of clumpy, kind of goopy. But that's it. That's Miss Enfield's DNA. So I hope you guys enjoyed this experiment. If you're in class, you got to do it in class. So this is mainly for DLI students or if you're absent. So if you guys have any questions, let me know. And then all of the supplies, if you guys have them at home, go ahead and try out the experiment and then see if you can see your own DNA. Let me know if you guys have questions and have a wonderful rest of your day. Thanks for listening. Bye.